Hi everybody, my name is Alessio Cavallari and I'm a postdoc at EAVAC in the Department of Environmental Microbiology. For the past 5 years I've been working in the LECO project, and today I'm going to be talking to you about the ecology of Legionella in engineered aquatic systems. As you might know, the LECO project is a multidisciplinary challenge. Researchers at EAVAC try to tackle this challenge in different work packages. I was responsible for work package 7, which deals with Legionella ecology. If you look at the simplified schematics of a plumbing system, you can see the water is flowing inside our pipes, it feeds either our shower, our taps, and you can see how bacteria are also living inside our pipes. These bacteria are organized in either a biofilm, which is a structure composed by bacteria and extracellular matter, but also planktonic, so swimming in the water. Now, some of these bacteria are pathogens and are contaminants of the water, for example, E. coli. We don't want this bacteria in our water. There are also pathogens which are non-contaminants and are indigenous in the water, so they live there. This is their environment. And a good example of this is bacteria belonging to the Legionella species. But why do we really care about Legionella? I'm not going to go super in details. There are other videos which I invite you warmly to watch that deal exactly on why Legionella is a problem. But just to give you a couple of facts, so you can get infected if you inhale Legionella containing aerosol. Legionella enters our macrophages, these are cells in our lungs. They have some escape mechanisms so they don't get degraded, and they produce a lot of effectors and replicate. The numbers of Legionnaire's disease have been increasing in the last 20 years. This is a graph which is relative to Switzerland, and it indicates the incidence per 100,000 population. That's a comparison with EU and USA in the same years. We know that there are many sources of infection, for example, cooling towers, and the main species within the genus that is responsible for infection is Legionella pneumophila, but we know that the genus Legionella comprises more than 70 species, and about a half are considered up to date clinically irrelevant. Mitigation strategies that are currently in place, they involve the use of temperature and disinfection, but those are not always effective, and Legionella often regrows in engineered aquatic systems. I just want to point out, when I say more than 70 species, about a half relevant, this is important for this presentation. Let's go back to our schematics of a plumbing system. We can see how Legionella is part of water microbial communities, and they engage in all sorts of interactions with other microorganisms. So they, as said, they can replicate intracellularly in protists, but these protists can also sometimes graze on Legionella, and so they can degrade them. There are other bacteria in the biofilm that have direct inhibition mechanisms, for example, the use of biosurfactants that can kill Legionella, and obviously because they live in the same ecological niches, Legionella compete with other microorganisms for nutrients. There are also examples of positive interactions, so bacteria, for example, cross-feeding for amino acids, so that both can survive. So Legionella, as said, have a biphasic lifestyle, so they can live intracellular and extracellular. And the important point is, can we use such interactions to develop a biological control of Legionella in plumbing systems? The way we want to observe this is using two different approaches. One is a molecular-based approach. The other one is an inhibition-based approach with dedicated lab experiments. I'm going to be starting with the molecular part. And so the first experiments that we conducted were performed on a set of shower hoses, 85 shower hoses collected from the same building, same material, age, user profile. On these shower hoses we did biofilm extraction, digital droplet PCR, and sequencing. What you can already see is that Legionella species, despite the similarities in the shower hoses, displayed a very differential pattern of occurrence. And Legionella pneumophila is only a small portion of the total Legionella species that we detected in this shower. So this made us think maybe the microbiome, the surrounding microorganisms are influencing this occurrence of Legionella. So in order to observe this, we performed statistical correlations to see whether bacteria, but also protists, could predict the relative abundance of Legionella. What you can see from these two plots here is that for 16s, so for prokaryotes, a lot of positive associations, a lot of bacteria are predicting high Legionella relative abundance, only a few are predicting low Legionella relative abundance. While for protists, the situation is mixed. We have protists that predict higher Legionella relative abundance and low Legionella relative abundance. This was done together with Emil, who helped me with the statistical analysis, and he also was part of the LECO project. So we see that overall we have a prevalence of positive associations, and the presence of positive and negative associations with protists can be interpreted relative to the different possible scenarios in the environment. Sometimes Legionella is digested by the protist, sometimes they replicate inside the protist and then they are released in the environment, sometimes they just seek protection with no replication, and so nothing really happens. So the different outcomes in terms of predictive abundance can be due to this different possible scenarios in the environment. These associations, of course, are not spatially resolved. This means that we might be seeing associations of bacteria that never really have an encounter in the shower hoses, just because we're not looking at the spatially resolved sample. The diversity of Legionella is what also caught our attention. So often statistical and correlation analysis are conducted at the genus level, meaning that we're looking at how protists but also bacteria are correlating with the genus Legionella in general. But we know, I told you this before, Legionella is made out of more than 70 species, and understanding how these associations can be resolved at a species specific level is very important. So we saw Legionella pneumophilia only represents part of the total Legionella species in our samples, and so this means that multiple species of Legionella are co-occurring in the biofilm. What we found was 30 amplicon sequence variances. You need to see these amplicon sequence variances as 30 different type of Legionella that we have in our samples, and they were assigned to the genus Legionella. So you see, from looking at only one organism, we are now looking at 30 different organisms, and what we tried to do was resolving these associations relative to the individual amplicon sequence variances. And what you can see, this is just a schematic of our findings, but you can see how we detected positive, negative correlations of Legionella and other microorganisms, or Legionella between each other. So this indicates that possibly different Legionella species in the environment are colonizing distinct ecological niches, which is important from an ecological. This is the actual figure that was published in the article that we wrote. And so what do these interactions tell us? Again, several Legionella species are occurring in biofilm. They possibly occupy different ecological niches. On the other side, we also looked at the possible inhibition that some aquatic bacteria can display towards Legionella species. Before starting, I want to mention that this work was conducted in collaboration with 4 excellent bachelor students that were embedded in the LECO project. So Max, Josh, Oliver, and Tobias helped me develop these experiments. What we really did here was screening more than 200 aquatic bacteria isolates against one reference strain of Legionella pneumophilia. These active isolates were then screened against multiple Legionella species. Here you can see a bubble plot. This indicates you when an inhibition occurred. You see a bubble of a different color and size. When no inhibition occurred, you see a black spot. What you can immediately see here is that this inhibition is different. There were only 3 isolates. They turned out to be the same species after all. They can inhibit all Legionella species tested in this work, while the others were inhibiting only some of them, and to a certain degree. Pseudomonas lurida, which is an environmental bacteria often found in water, could inhibit all the species of Legionella that we tested. And so we thought that this was really interesting. But at the same time, we see also that species-specific inhibition patterns are not always consistent. We see two species of Remundimonas aurantiaca that are in one case inhibiting almost all the Legionella species tested, in the other case only two of them. And we also identified for the first time some bacteria that are linked to Legionella inhibition, for example, Pseudomonas lundensis. On the left-hand side of this slide, you can see how a classic inhibition experiment looked like. We have, this is called a spot-on-loan assay, where you can see this is an inhibition area. It can be either big, like the one indicated here in this figure. This one is the Legionella loan underneath, and you can see where the arrow is indicating there is no inhibition in this plate. Sometimes the inhibition can be just, restricted or limited to a certain area. Next, we were intrigued by what compounds could actually be responsible for this inhibition. And so we screened the genomes of the antagonistic bacteria for the presence of secondary metabolites, of gene clusters related to secondary metabolites that could explain the inhibition. This is a work that was done with collaboration with Serena Robinson's group at EAVAC, who was of a big help. What we found was different classes of compounds, starting with lipopeptides. Those are biosurfactant. Their main mode of action is related to the polarization of the cell membrane. Among those, we find bananamide, rhizomide. We also find siderophores. They are iron-chelating molecules. We know that Legionella needs iron in order to grow. Lipodesipeptides, those are a class of lipopeptides which is mainly produced by the genus Pseudomonas, and among those we find viscosine and tolacine. Lactopeptides, which are ribosomally synthesized and post-translationally modified peptides. They are known to have antimicrobial activity, but we could not link any of these gene clusters with an actual name in our dataset. What is really interesting here is the presence of viscosine, which was only found in the genome of those three strains with the broadest antagonistic ability towards all the Legionella species tested in this study. This got us curious, and so we tried to see whether we could find viscosine actually in the total extract from a co-culture of Legionella, any antagonist after HPLC and fractionations. The different fractions which were prepared through HPLC were tested for the inhibition of Legionella. This was done in collaboration with Silke, who is a member of Serena's Robinson group. Here you can see TIC and EIC from the co-culture extract, and you can see that there are peaks that correspond to the mass of viscosine, so we could confirm that viscosine is actually part of this extract, so it's produced by the antagonistic bacteria. And when we fractionated the co-culture, we noticed that the different fractions also had antagonistic activity towards the Legionella species. This is an MS-MS spectrum that confirmed the structure of viscosine, which is actually present in our extract. But what do these interactions tell us? So they tell us that different Legionella species' susceptibility to antagonistic activity is variable. This is reflected maybe in the system they use disinfection, and that's why maybe some species are always surviving after treatment is applied. But from an ecological point of view, variable interactions maybe indicate, again, the occupation of separate ecological niches. These observations, so the one described in the first part of this presentation and the ones related to the inhibition of Legionella, got us thinking. And together with Marco, we started investigating the diversity of Legionella. Marco conducted a study that investigated the differences in all available genome of Legionella with ecological implications. And this is part of a different video, which I warmly invite you to watch in this LECO series. And this work also led us to imagine an application of a potential probiotic approach. This is now part of an InnoSwiss funded project where we will try to bridge towards an application of these results. This is a schematic of the proposed approach where we will use existing filters which we will preload with antagonistic bacteria in order to seed a system with bacteria that are antagonistic to Legionella, trying to prevent their occurrence. Our target is, at least for now, new buildings. And with this, I will thank, I'm at the end of my presentation. I'd like to acknowledge Frederick Hammes, Antonia Heichelberg, who run this project throughout the years, and all the people involved in this work. Obviously, the Drinking Water Microbiology Group, of which I'm part, and the LECO Consortium. I thank you for your attention. If you'd like to have more information about my work, you can contact me. My email address is on this slide, and I warmly invite you to watch other LECO videos, which are very interesting and will tell you more about Legionella in plumbing systems. Thank you very much.